Okay, this is the chapter on photosynthesis, and as it says here, photosynthesis is the process that converts solar energy into electric or chemical energy. So in photosynthesis, energy from the sun, from light, is going to be captured by plants, and through a process called photosynthesis, that energy is going to be stored in the bonds of complex carbohydrates that, when we ingest those, we can break the bonds and acquire energy. And we can also ingest other heterotrophs and acquire energy that they've processed or they've acquired through eating other plants, plants and or other heterotrophs. It says directly or indirectly, photosynthesis nourishes almost the entire world. Yeah, all the part that's exposed to light. We'll see in this chapter that there are some that do not acquire their energy from light. So we'll look at that in just a little bit. Definitions of autotrophs, if you remember that, autotroph means self-feeder. Autotrophs and producers are the same thing. They're green plants, and they most of them use energy from the sun to trap in bonds of complex carbohydrates. Heterotrophs means other feeders. Consumers are heterotrophs. They cannot make their own energy. They have to acquire it from plants and other heterotrophs. Now, autotrophic nutrition, the self-feeders, there are two types. Photoautotrophs are ones that use the energy from the sun, like photoautotrophs will be your producers or your autotrophs, and they are going to capture energy from the sun, again, and store it in complex carbohydrates. So the source of energy for photoautotrophs is light from the sun. Chemoautotrophs are another type of autotroph. Now these do not capture light energy. These are going to be like the ones that are down at the bottom of the ocean where there's no light, so they can't photosynthesize. But what they do is they're next to those volcanic vents, and so they have gas bubbling out all the time, and that's hydrogen sulfide gas. Well, chemoautotrophs can break down hydrogen sulfide to acquire their energy needs, and they grow and they can produce a food web at the bottom of the ocean where there's no light that penetrates that deep. And sometimes you'll see that on a National Geographic or something. They'll show a food web down right next to a volcanic vent. Heterotrophic nutrition, remember I told you heterotrophs are the same thing as consumers, and they feed off of autotrophs and other heterotrophs. Now, decomposers are a special type of heterotroph. Decomposers are fungi, and they feed off of dead material and return the elements of that biomass to the environment to be recycled. This is showing about photosynthesis being endergonic. That means it's going to capture energy and to store it into the bonds of glucose. Now you see over here on the far left, there's carbon dioxide and water. Those are the raw materials for photosynthesis. The raw materials for photosynthesis are carbon dioxide and water. This yellow line represents energy from the sun that is captured by photosynthesis, and it's going to be stored in the bonds of glucose, which will be converted to starches and other molecules in the plant. So energy is stored. It's an endergonic process. Here's an overview of the equation. This is about as short as you can get. And you see on the far left, it says raw materials we produce in our cellular respiration. When we exhale, these products come out. These are waste materials from our own cellular metabolism of our cells. Carbon dioxide gas, water. Well, those happen to be the raw materials for photosynthesis. And so plants will use carbon dioxide and water to produce this arrow here represents through photosynthesis, produces the products. First one is glucose, C6H12O6, that's glucose, plus oxygen, plus water. So those are the products of photosynthesis. Glucose, and that can be converted to starches in plants, oxygen, and water. Now previously, the scientists believed that carbon dioxide, the gas that we exhale in our waste material, is what plants would use that break down the carbon dioxide and return the oxygen to the environment. Well, a man named C.B. Van Niel said that he thought it was not carbon dioxide, but it was water. And the scientists said, well, prove it. Well, they had not even proven that it was carbon dioxide, so they're kind of a hypocrite when it comes to that. But they made him prove that it was water, and he did. What he did was he labeled the oxygen of water with an isotope, O18. It's an isotope of oxygen. And he watered these plants with the isotope labeled water. And then he analyzed the atmosphere and found out that the atmosphere had the isotope of oxygen in it. Because of that, water was broken down, and it was where oxygen comes from when plants produce oxygen. So he was vindicated by his experiment proving that water is actually broken down into oxygen that's released by the plants, not carbon dioxide. Now we have a redox process. Redox is oxidation and reduction, and that's going to imply an electron transport chain or electron transport system. And plants have those. Plants have electron transport chains, as well as they have a chemiosmotic apparatus, just like animal cells do. Remember the mitochondrion? So they have this, and it's located in a similar area we'll look at. Photosynthesis also has two parts. The first part is called light reactions. This is the light-dependent phase of photosynthesis. So part of photosynthesis requires light and part doesn't. This part does. And you see it's called photophosphorylation. Phosphorylation is the addition of a phosphate molecule to another molecule, addition of a phosphate to another molecule. And the energy from light is used to do that. So this is going to make ATP, is what's going to happen. The phosphate is going to be added back to ADP to make ATP. Now the Calvin cycle does not require light. We used to call it the dark reactions. But the Calvin cycle does not use light. This is where carbon fixation occurs. Now the light reactions, that's going to use water. Calvin cycle, that uses carbon dioxide. The two raw materials for photosynthesis are used independently. Oh, here's the summary equation again. It's missing the arrow there. There's your raw materials, carbon dioxide plus water produces. This is going to be glucose plus oxygen plus water. Okay, so the location of photosynthesis is in the chloroplast. The chloroplast is the site of photosynthesis in plants. This is where it says the essence of photosynthesis. This means what is required for photosynthesis to occur in these plants. One thing for sure, that the plant has to have chlorophyll A. It has to have chlorophyll A. That's the essential photosynthetic pigment. It's got to have that one. And the accessory pigments, we'll look at probably three more. They're going to be chlorophyll B, one called carotene, C-A-R-O-T-E-N-E. That's going to be an orange pigment. And then xanthophyll, which is a yellow. It's X-A-N-T-H-O-P-H-Y-L-L, xanthophyll. There'll be two photosystems, photosystem 1 and photosystem 2. And we'll look at those as we go through this process. Carbon dioxide and water, the raw materials that are used, and you got to have light. And the source of light outside is going to be the sun. Here's a leaf, just to let you know what a leaf looks like. As it says over there, the leaf is the organ of photosynthesis in most plants. Because some plants don't have really what you would call a leaf, they have stems, like a stick plant. And the chloroplasts are in the stem. On this one, not only are chloroplasts in the leaf blade itself, but they're also in the petiole, the little stem here. And so photosynthesis can occur in both areas. So reading here, it says the blade is the flattened portion of the leaf that collects light energy for photosynthesis. This is a leaf blade from a tree. Well, grass blades are also blades, and they have chloroplast in them, and they photosynthesize also. Leaves come in a variety of shapes. Most photosynthesis occurs in the mesophyll of the leaf. Now we're going to look at where that's talking about. There are two mesophylls. We're going to look at the palisade and the spongy mesophyll. And the palisade mesophyll is where we're talking about here. Most of it occurs there because the spongy mesophyll does not have any chloroplast in it. So let's look at a cross-section of a leaf. Here's the cross-section. Starting off at the top and the bottom, here's the upper epidermis at the top and the lower epidermis at the bottom. Neither one of those has chloroplast in them. They are there to produce this little red waxy layer on the surface to help to waterproof the leaf so it doesn't lose moisture very easily through the epithelial cells, or the epidermal cells, I mean. Now, you can see, look at the bottom lower epidermis here, and look on the far left, you see stomata and guard cells. Stomata are openings that allow gas exchange and water exchange, partial water, water vapor, between the atmosphere and the leaf. And the guard cells are the cells that will open or close the stomata. If you get your hands and put them together like you're praying, and you look at your index fingers, and then you bend your index fingers, it makes a hole. The hole would be the stomata, and your fingers would be the guard cells. They can open the stomata or they can close the stomata. And you see that they're opening into a mesophyll. Look on the far left, spongy mesophyll. This is real open, and so gases can be exchanged here, and so can water vapor between the leaf and the environment. And above that is the palisade mesophyll. Palisade, these are stacked like cigars. You see little green dots in them. Those little green dots are the chloroplast. So most of your photosynthesis is going to occur in the palisade mesophyll. Now we're going to look at something else in just a minute. Look at the bottom, the middle word down here, bundle sheath cells. Now this bundle is opened up, so you can see the darker material in the middle. That's going to be xylem and phloem. But these cells that form a ring around the xylem and phloem, those are the bundle sheath cells. You can see one over here, bundle sheaths. Those also have chloroplast in them, and we'll see what their function is a little bit later. The xylem and phloem have to do with movement of water and food materials, minerals up and down the leaf in the plant. You get that in 1407. Nature of sunlight. Well, sunlight is electromagnetic radiation. I got energy here, electromagnetic radiation. And we can only visibly see part of the electromagnetic spectrum. So we'll get to that in just a minute. What travels in waves? Electromagnetic energy does travel in waves, and we'll see the different forms of electromagnetic energy on the next slide. We can see the visible spectrum. We see visible light. This is only a small part of the electromagnetic spectrum. The visible light we see, the units of light are called photons. Photons are infinitesimally small, and they do have mass. So they do exist. They are forms of matter, but they are extremely small and lightweight. But those are the units of light. They're called photons. The photosynthetic pigments that I talked about earlier, the ones that we were going to get into, chlorophyll A is essential for photosynthesis. And then the other ones will vary. Chlorophyll B is also another pigment for photosynthesis. And the carotenoids. This is a group. Carotenoids are a group of yellow and orange pigments. And like I told you a minute ago, carotene and xanthophyll are carotenoids. So they're in that group of yellow and orange pigments. And those are the only ones that we'll really look at: chlorophyll A, chlorophyll B, carotene, and xanthophyll. Now here's the electromagnetic spectrum here. And you see that the visible light spectrum is the one that we see in. And what we can't see, ultraviolet. This is what typically sunburns us outside. So, that's not real good to have too much sunlight. A little bit's good for vitamin D production by your skin, but not a whole lot. X-rays, those can be bad too in high levels, but they use X-rays for dental, and dental and X-ray techs use those. Gamma rays, those are bad. Those are so high energy and so small they can shred your DNA, and those are used to sterilize things with, but they can kill you very easily by shredding your DNA. It can't repair itself. Infrared, we use those for TV controls. You can look at your TV control and flash it, push the buttons and you won't see anything because we don't have receptors for infrared light. But your TV and your video players do. Microwaves for cooking. They're longer wavelengths and the radio waves are even longer. So we see right kind of in the middle of the spectrum. It's called the visible light band or spectrum here. And you see that it's listed out here like a prism, all the colors that make up the visible light spectrum. The red end, these are longer wavelengths, these 750 nanometers roughly, longer and lower. I always kept the L's, longer and lower, told me that that's what the red was. On the opposite side, the violet, shorter wavelength, higher energy. Well, you can see that it gets higher and higher and higher until it's destructive, or lower and lower and lower. It's not so destructive, but that still is not good for you. So you probably in middle school, high school, I don't know when they told you how to remember the colors of the rainbow by a person's name, Roy G. Biv. Red, orange, yellow, green, blue, indigo, violet. Roy G. Biv keeps them in order. It goes backwards on the scale when you're looking at it, but that's okay. At least you know what the order is. It's like a rainbow. It's in the same order. So the red end, longer wavelength, lower energy. All the way to the violet end, shorter wavelength, higher energy. And that's the visible light spectrum that we see. And plants will use portions of this visible spectrum for photosynthesis. And we'll see that on the next slide. On this slide, you can see, here's chlorophyll A up here. And you see the spectrum is going all the way across. Well, let's see what chlorophyll A does. It's absorbing way over here in the 400s. It kind of peaks up, and as it gets toward the blue-green or green, it kind of tanks. Hardly anything at all. And as you get back over here, chlorophyll A peaks a little bit in the red zone. So in the red wavelength, it uses a little bit of red. Blue, indigo, violet's what it uses, and a small portion of the red spectrum. Well, let's look at chlorophyll B. Chlorophyll B, it's in the blue, indigo, violet also. There it is, real high absorber, and then it drops off when it hits the green also. And it comes back over here, and it peaks just a little bit in the red spectrum. So the chlorophyll A and B only use a small portion of the red. They don't use it all, just a little bit. But they tend to use the blue, indigo, violet for sure. Your carotenoids, your xanthophylls and carotene, you see it uses blue, indigo, violet. And as it starts to get toward the green, it tanks and you don't see it again. They're not the ones that we're looking at do not absorb anymore. So getting rid of most of the color stuff on here, let's look at the next slide and it'll show you how plants absorb mainly in the blue, indigo, violet and a part of the red spectrum, which is why you see plants usually as being green or yellow. They don't use those links. Those are reflected. That's why the plants are green, yellow, orangey. Plants don't use those wavelengths. They're reflected. The ones that they do use are absorbed. So the blue, indigo, violet is absorbed, and part of the red is absorbed. But the greens, yellows, and oranges pretty much are all reflected. They're not absorbed. So most of the energy that plants get from sunlight is from the blue, indigo, violet, and part of the red visible spectrum. Okay, now this is fluorescence here, and you can see on the right hand side there's a little beaker there. It's showing red, and what's happened here is sunlight has been shining on these chlorophyll molecules, and as it shines on them, the electrons are excited and they jump from pigment to pigment to pigment, eventually go into the center chlorophyll molecule. It's going to be a special chlorophyll A, which sends the electrons to a high energy electron acceptor, and they start going through the electron transport chain, and as they do that, they're going to release that energy in the form of light, and you see it's in the red spectrum here, and the electrons are returning to the system to be excited again, And as they collect energy from the sun, they're excited, sent to high energy electron acceptor, go through the electron transport chain and lose their energy again, and in the form of this red spectrum here. They also lose heat too, but it's hard to really pick that up without special devices. So this is showing that if I had a green test tube of chlorophyll pigment extract and I shined a light through it, it would be green above and below where that light is shining through that tube. Where the light shines through that tube, the electrons are being excited, going from pigment to pigment to pigment, going to high energy levels, being sent down through electron transport chain, and they're going to give off light in the wavelength of a red spectrum. It's really kind of an orangey red, real light orangey red. So you'd see green chlorophyll pigment extract above the beam and below the beam, and where the beam of light's going through, it's not green. It's going to be a light reddish orangey color from fluorescence. Here are two pictures. The one on the left is a transmission electron micrograph of a chloroplast, and you can see the shape of the chloroplast, and you can see several things that are pointed out. The stroma is the fluid of a chloroplast. The chloroplast, look at the one on the right. The chloroplast has three membranes. An outer membrane, this light blue shell on the outside, that's the membrane. An inner membrane, this orange colored part of the picture. So it's got two membranes, but it has a third membrane. These thylakoid discs here, these thylakoid discs are a third membrane system of the chloroplast. And we'll see that these individual lines on the left, you can see little bitty skinny little lines. Those are individual thylakoid discs. But you see the word grana, it's a stack of discs. So look over here on the right hand side, third word down, a granum. Granum is a singular, grana is plural. So you can see it has three discs here. They're stacked together. And up above, there's three discs. So you see stacks all over the place. Well, that's what the dense structures are on this electron micrograph. Those are all grana, stacks of thylakoid discs. The stroma is going to be the fluid. It's going to surround these thylakoid discs. And you can see on the left-hand picture it's kind of light gray. And the right-hand picture, they're just kind of pointing out where it is. Some more terms. An antenna complex is a collection of photosynthetic pigments. And they call it that because usually they'll draw them like a dish antenna, circular. And it has all these pigments in a circle. And these are photosynthetic pigments like chlorophyll B, carotenes, and xanthophylls. And then the reaction center is in the center of that antenna complex, and it's a special chlorophyll molecule. It's a chlorophyll A. That's the one I told you is essential for photosynthesis. So in the center is going to be a chlorophyll A. Well, there are two photosystems, and each one has a different chlorophyll A pigment. Photosystem 1, in the reaction center, there's a chlorophyll A that's a P700 chlorophyll A pigment. So it has a special absorption potential, but it's going to be in photosystem 1. It's called a P700. Well, there's a second photosystem, photosystem 2, and it has an antenna complex also, but its reaction center has a little bit different chlorophyll. It's a P680 chlorophyll A molecule is in the center of photosystem 2. So the two photosystems right off the bat, they both have antenna complex, they both have reaction centers. Photosystem 1, the reaction center has a P700 chlorophyll A. Photosystem 2 has a P680 chlorophyll A. Now we're going to learn some other differences in these in just a minute. So that's the first two differences. Cyclic electron flow. Now cyclic means something happens over and over again, so something returns. Well, what returns? Well, the electrons return. So the electrons are going to be cycled and they return. It says or cyclic photophosphorylation. All right, so again, this is going to be, using light to phosphorylate an ADP to make ATP, and the electrons are cyclic, so they return. So they return to the system. And we're going to see a picture of that in just a second. Now, Photosystem 1, working by itself, produces ATP only. So these photosystems can work by themselves or with each other. At least photosystem 1 can. Photosystem 1, working by itself, produces only ATP, and that's going to be cyclic electron flow or cyclic photophosphorylation. If you look at this one, this is photosystem 1. And you see that the photons here, energy from light, are going to excite electrons, and they're bouncing from pigment to pigment to pigment in this antenna complex, and eventually are sent to the reaction center, P700. Photosystem 1, that's an odd number, so 7, so P700. P700 chlorophyll A sends them to a high energy electron acceptor, and they go down through the electron transport chain, and they lose energy that's captured by the cell to be used to make ATP, but you see they return to the system, and they're excited again, sent to the high energy electron acceptor, go down through the electron transport chain, lose their energy, and return again. Well, the energy that's lost is captured, some of it's captured by the cell and used to make ATP, look at that, by chemiosmosis. So we're going to talk about that. It's very similar to what happens in the mitochondrion. So photosystem 1 is cyclic electron flow or cyclic photophosphorylation because the electrons return to the system. Photosystem 1, working by itself, only produces ATP. That's it. And let's look what happens to that with that energy. High energy electrons are sent to the electron transport chain. That's what I just told you on the previous slide. The electrons from photosystem 1 are excited by photons, light, and transferred to electron transport pigments, which release the free energy. These electrons are returned. All right, so the energy. It's released by these high energy electrons as they went through the electron transport chain and returned to the system. The energy is used to pump hydrogen ions into the thylakoid space. Those thylakoid discs are hollow, and there's a space inside. So the energy that the electrons lose, or that the cell captures, is used to pump hydrogen ions into the thylakoid space. Here's my thylakoid disc here. It's kind of a real simple disc drawing. Setting up a gradient to produce ATP. So you see my hydrogen ions are being sent or pumped into the thylakoid disc. If you look at here, 1,000 times greater hydrogen ion concentration inside than outside. So I put the pH difference. pH of 8 outside, pH of 5 inside. So it's pretty acidic inside. We're going to see in a little bit that right here, where it has ATP synthase, there is a chemiosmotic apparatus right there in the membrane of the thylakoid disc. And so hydrogen ions can zip through that chemiosmotic apparatus in the presence of ATP synthase, and ADP is rephosphorylated to make ATP. So we'll see that again. Now, if there was a cyclic, there's also a non-cyclic electron flow or non-cyclic photophosphorylation. That means that when the electrons go down the electron transport chain, they do not return. So they're lost, and the electrons have to be acquired from somewhere else. And we'll see where in just a second. So it's our next slide. Now, non-cyclic electron flow or non-cyclic photophosphorylation produces three things: ATP, NADPH plus H, or H2, they never really put that, but see, this NADP is the electron and hydrogen carrier. You could think of P for plants, just that to help you key that. So plants have NADP that pick up 2 electrons and 2 hydrogens. And it also produces oxygen, which is released to the environment, which is how we know plants produce oxygen. Now, the non-cyclic photophosphorylation involves photosystem 1 and 2. When 1 is tied to 2, it is no longer cyclic. Okay, working by itself, photosystem 1 is cyclic. But when it's tied to 2, it is no longer cyclic, and we'll see why on the next slide. Okay, here's photosystem 2. In photosystem 2, on the left here, let's look at some stuff over here. What's happening here? In photosystem 2, you see that you have light, and you see these exciting electrons, and they're bouncing from pigment to pigment to pigment, this black line with the arrow, and eventually reaches the reaction center, P680. Now 6 and 8 are even, so is the number 2 for photosystem 2. So the P680 is the chlorophyll pigment A in photosystem 2. That chlorophyll A is going to send the electrons to high energy electron acceptor, and they're going to go down through the electron transport chain and release energy that's used, you can see, to produce ATP by chemiosmosis. So that energy is going to be used to pump hydrogens into the thylakoid disc, and then as they escape through the chemiosmotic apparatus, they make ATP. Well, these electrons don't return. They're sent over to photosystem 1, which it also is going to have light shining on it, and the electrons are going to be absorbing energy and bouncing from pigment to pigment to pigment, and they eventually reach the reaction center with the chlorophyll A of P700 chlorophyll A, which sends them to high energy electron acceptor, and they can start going through electron transport chain, but look what picks them up. NADP comes by and picks up the electrons and hydrogens to make NADPH plus H. Okay, so let's look way back over here on the left. If those electrons were lost, how does it acquire new electrons? Look at number 2 here. Splitting of water releases oxygen. So when water is broken down, oxygen is released, and that's what the plant produces that we breathe. It also produces 2 hydrogens and 2 electrons. And those electrons are the ones that are replacing the ones that have been lost. So water is constantly being broken down, and water is the source of the oxygen that plants produce, as well as the electrons that keep being lost in this non-cyclic photophosphorylation. So water is the source of electrons, and water is the source of oxygen. So let's go through that again. Remember, there were 2 raw materials for photosynthesis, water and carbon dioxide. Water is used for the light-dependent phases. So here it is. Water is right here. Water is the source of oxygen and electrons. So water is broken down, oxygen is released to the environment, and the electrons enter this photosystem 2. Light excites those electrons. They're bounced from pigment to pigment to pigment until they eventually reach the chlorophyll A, that's a P680 chlorophyll A, which sends them to high energy electron acceptor. They go down the electron transport chain, giving up their energy, which the cell captures and uses to pump hydrogen ions into the thylakoid disc, and they escape through the chemiosmotic apparatus to make ATP. Well, those electrons are transferred over here to, which in turn it will excite the electrons and then move from pigment to pigment to pigment, and eventually reach the P700 chlorophyll A in the center of the photosystem 1. They're sent to high energy electron acceptor and start going down the electron transport chain again, but they're picked up by NADP to make NADPH plus H. If you remember, the products of the non-cyclic photophosphorylation are ATP, NADPH plus H, and oxygen. Now here's just some information about this non-cyclic photophosphorylation. Like I was telling you earlier, the electrons do not return to the chlorophyll from which they originated. Okay, they don't return. Remember, photosystem 2 lost them. So that is non-cyclic. The products, ATP, NADPH plus H, or H2, and oxygen are produced. Okay. Electrons from photosystem 1 reduce NADP to NADPH plus H. Electrons of photosystem 1 are replaced by electrons from photosystem 2. That's how it keeps getting its new electrons. The energy from the electrons of photosystem 2 result in the production of ATP through chemiosmosis. Remember, when the electrons are going down the electron transport chain, that energy is being used to pump hydrogens into the thylakoid disc, and then they escape the disc through the chemiosmotic apparatus with ATP synthase to make ATP. As water is split, electrons from water replace those lost from photosystem 2. This also results in the production of oxygen and hydrogen ions. These hydrogens are what the NADP is picking up. This is showing the thylakoid disc membrane here on this one. And you see that this, up here at the top is your photosystem 2 and your photosystem 1. So it's showing how water is used to supply the oxygen and the electrons. Light's exciting those electrons, and as they go through electron transport chain, they're going to pump hydrogens into this thylakoid disc in here, which is kind of like light brown. And they're sent to photosystem 1. The electrons are, and they start going through electron transport chain again, but NADP picks them up to make NADPH plus H. The hydrogens in this thylakoid disc, this brown area, you see are going through the chemiosmotic apparatus in the presence of ATP synthase, and enough energy is produced to put that phosphate back on ADP to make ATP. Now, just some things to think about. Remember the products of this non-cyclic photophosphorylation were oxygen, which you can see over here on the left, that's released by the plant. But the ATP and the NADPH plus H are sent to the Calvin cycle. They have to be used by the Calvin cycle. The Calvin cycle has no way to get energy, so the light-dependent phase made the ATP for it, and these hydrogens and electrons are going to be taken in by NADPH plus H. Now the Calvin cycle, also called the light-independent reactions or the dark reactions, this Calvin cycle does not use light. If light is on, that's fine. If light is off, that's fine also. It doesn't use light. So the Calvin cycle doesn't use it. Where is the Calvin cycle? It's in the stroma of the chloroplast. So the light-dependent phase, the cyclic photophosphorylation, that occurs in the thylakoid disc membrane, the membrane of the thylakoid disc. The Calvin cycle is in the stroma, the fluid part of the chloroplast. Carbon dioxide, the second raw material for photosynthesis, is reduced, that means it's going to accept electrons, by NADPH plus H. Remember, it's carrying electrons and hydrogens. ATP from the light-dependent phase provides the energy for the processes to take place. And the products of the Calvin cycle, carbohydrate and water, are produced. And the carbohydrate is going to be, we'll see what that is in just a minute, it's going to be converted to glucose and then to starches. Here's a picture of the chloroplast again, a transmission electron micrograph on the left and a drawing on the right. And again, you can see, let's look at the bottom here. It says outer membrane, which is light blue, the inner membrane, which is the orange, and the third membrane, the thylakoid disc on the inside. You can see they're green. You also see on this picture, you can tell that they're hollow there again. Stacks of thylakoid discs are called grana, and a single one there is a granum. Over here on the left, you see the stroma. On the right, you see the stroma also, just pointing to the inside. To the left, you see the grana, the stacks of thylakoid discs, and you can see individual thylakoid discs, the little thin lines. Here's the Calvin cycle, and don't get too freaked out by this. We're just getting the overall view of it here. Now you see it has 3 molecules of carbon dioxide coming in. Remember, carbon dioxide is a raw material, and that has 3 carbons, right? So keep that in mind. As it enters, there's an enzyme called rubisco that it will bind to, and it allows that to bind the carbon dioxide to this ribulose bisphosphate molecule. It says 3 molecules, and there are 5 carbons each. So it's 3 molecules of carbon dioxide, 3 molecules of ribulose bisphosphate, and you see that it makes 6 molecules, which are this is called 3-phosphoglycerate. I'm not going to ask you that, but you can see that there are 3 carbons big. So when this 5-carbon molecule and this 1-carbon molecule went together, it was an unstable molecule, and so it split into 6 3-carbon molecules. ATP is used. That was produced by the light-dependent phase. NADPH is used. That was by the light-dependent phase. And at the bottom, you see that there's 6 molecules of glyceraldehyde phosphate produced. Well, the cell harvests 1 of those molecules per turn of this Calvin cycle. Every turn, the cell gets to harvest 1 molecule of glyceraldehyde phosphate. And you see that it is 3 molecules. 3 carbons. Remember, there were 3 molecules of carbon dioxide that came in. That was 3 carbons. This glyceraldehyde phosphate is also 3 carbons. So glyceraldehyde phosphate is going to be used to make carbohydrates, and usually those are starches. So per turn of the Calvin cycle, when carbon dioxide enters the Calvin cycle, per turn, 1 molecule of glyceraldehyde phosphate is harvested, which will be converted to glucose and starches. Now there's a problem. It's called photorespiration, and that problem can occur where there's light and warm temperatures, just like normal, but oxygen will compete with carbon dioxide for the binding site on that ribulose bisphosphate carboxylase, which we abbreviated as Rubisco, a little easier to say. Now the reason it does that is because, remember we talked about mimics, where they're similarly shaped? Well, here's 2 oxygens, here's 2 oxygens. It's going to be a similar shape in those molecules, and so oxygen can bind to Rubisco just like carbon dioxide can bind to Rubisco. And so it's competitive inhibition. Oxygen's competing for Rubisco. Well, Rubisco is supposed to bind to carbon dioxide. So there's a problem. It's called photorespiration. The Rubisco uses oxygen and releases the carbon dioxide. This ribulose bisphosphate, that 5-carbon molecule, is oxidized, thus reducing the photosynthetic output up to 50%. So it's about a 50-50 competition between oxygen and carbon dioxide for this enzyme. And so it reduces the photosynthetic output. Oxygen competing, that competitive inhibition reduces the productivity of that enzyme. Well, the solution, there's a solution. The plant has a way to get around that. That happens and the plant will live with it. Photorespiration happens. But the plant has a way to get around photorespiration and a part of the plant itself. It's called C4 photosynthesis as a solution to photorespiration. In C4 photosynthesis, the carbon dioxide is pumped from the mesophyll to the bundle sheath cells as an organic acid. So it is actually harvested from the spongy mesophyll, bound to another molecule, and sent into the bundle sheath cells. Malate releases the carbon dioxide. Once it's in there, malate is malic acid, releases the carbon dioxide in the bundle sheath cells where normal photosynthesis, C3 photosynthesis, is completed. So let's look at the next little picture here. And you can see that here's carbon dioxide coming in from the atmosphere through the stoma. Here's your 2 guard cells. Here's the spongy mesophyll here. And it is bound to an acid. Here's pyruvic acid. Usually they just call it pyruvic. Carbon dioxide is bound. It makes malic acid. And the malic acid is sent into the bundle sheath cells, where it releases the carbon dioxide, becomes pyruvic acid, goes back out, picks up carbon dioxide, becomes malic acid again, comes back in, releases carbon dioxide, becomes pyruvic acid, and it keeps going around and around and around. And that carbon dioxide can go right in, bind to Rubisco, and complete this Calvin cycle to make glyceraldehyde phosphate. Oxygen can't get in here. The carbon dioxide is brought into these bundle sheath cells. Oxygen doesn't get in. So this is a way that the cell gets around photorespiration. It's called C4 photosynthesis. And so plants have these bundle sheath cells which are surrounding the xylem and phloem, and that's where the carbon dioxide is sent. No competition with oxygen then. Here are some bundle sheath cells. You can see these dark masses of green. They have a lot of chloroplast in there. And you see a whole bunch of bundles here. And in the middle of these bundles are xylem and phloem. Xylem and phloem are what carries water, minerals, and nutrients up and down the plant. So they're inside of these bundle sheath cells arrangement here. There's also a way that plants get around hot and dry conditions. Like the desert is called CAM metabolism, which stands for crassulacean acid metabolism. That's why they make shorthands like CAM, because it's a lot of words to say, and it's kind of weird sometimes, or hard to say. So CAM metabolism is how plants that live in like sunny, dry, warm areas like the desert. The desert's an area like that, and you know that that's hot and dry, and there's only a certain type of plant that can live out there, and usually it's a type of a cactus. So this allows the plants to conserve water, and it works just the opposite of most plants. The stomates, all the stomata, are closed during the day. Now the reason they're closed during the day, remember I told you that when they're open, carbon dioxide and water can be exchanged between the environment and the plant. If they were open during the day, water would be lost constantly to the dry atmosphere. It would go from the plant to the atmosphere, evaporation, essentially from the plant, it would dry it up. So during the day, the stoma are closed, so water loss is at a minimum, but they're open at night. Now, you know at nighttime, you have a lot of condensation. If you go out early in the morning to get your paper and it's in the grass is wet. Moisture has condensed out of the atmosphere. And so cactus open their stoma at night where there's more moisture in the atmosphere, and they don't have to worry about losing water. So at night, carbon dioxide is absorbed at night and converted to organic acids. So the organic acid amount builds up to high levels inside of the cells of the cactus during the night, because during the day the stoma are closed, and here it says organic acids are released. Release the carbon dioxide during the day. So the acids are sent to the Calvin cycle and release the carbon dioxide during the day. So at night the levels build up high, and during the day they're used up and they become lower and lower and lower as the day progresses. And at night they're built back up again. So the stoma are open and closed oppositely to plants like where we are right now. This is a little slide here showing the C4 and CAM photosynthesis here. You see here that the mesophyll cell has organic acids built up during the day, sent to the bundle sheath cells. Okay, so they're sent to the bundle sheath cells. That gets around photorespiration. And this is the CAM, crassulacean acid metabolism, like a cactus. At nighttime, carbon dioxide is converted to organic acids, and during the daytime, the acids are sent to the Calvin cycle to release the carbon dioxide to the Calvin cycle. So you can see both of those.